is going to be a little bit of, or there are multiple complexities to it. And in the beginning itself, I should mention that we will discuss this twice, probably, or at least I'll post another video again in case of an issue. I don't, how many of you have read the concept of genomic imprinting? Those who have studied stem cell biology must have read about it. So in today's class, what we are going to do is how to study the epigenetic status using molecular techniques. So the focus is about one particular technique, but the interest, my actual focus is that your ability to interpret the results. Okay, it has to do more of thinking rather than just the technical part. Not just the technical part, you'll have to use your logic and so on. So what is epigenetics? We'll have a quick introduction to what is epigenetics. I think I have already discussed this before, but I'll just go this. In epigenetics, there should be a change in gene expression, and the change in gene expression should be because of any mechanisms other than change in sequence. So if there was a change in sequence, it would have been a mutation. So any mechanisms on the chromosome, okay? So these mechanisms are predominantly, you will have DNA methylation, histone modification. Those are the well understood ones. And then we have chromatin modeling, and then we have nuclear architecture. So these are the four mechanisms, and in particular today we are going to study mostly about how to analyze DNA methylation. Okay, there are more techniques, but for this chapter we will, at this point we will only learn about that particular one. DNA methylation using restriction endonucleases and southern blotting. So we will use the concept of, we'll try to read the concept of genomic imprinting, which is also a type of epigenetics. So there are three things about genomic imprinting. First one is it should be an epigenetic mechanism. Okay, imprinting is an epigenetic mechanism. Second thing is it is monoallelic expression. Monoallelic expression. What is monoallelic? So if you have a gene X and we have gene Y, and assume that, this is gene X also. One is from the paternal allele. We have inherited this gene X from father, and the second one from the mother. If only one is expressed, monoallelic means both should not be expressed. Only one is expressed and the other one is repressed. That is called as monoallelic expression. And the third part of the part about genomic imprinting is the expression should be dependent on the parent of origin. Okay, parent of origin. I think I'll just say what it means. So we have two parents, right? One, we have a father, and one a mother. So this gene X is only expressed from paternal allele. Okay, and it is repressed on the maternal allele. What does it mean? You have expression. It is a monoallelic because there is only expression from one of the alleles, and we are saying all the time only paternal X is expressed, paternal X chromosome, gene X is expressed, maternal is repressed. To elaborate a little bit more, because I don't know, some of you may not have done stem cell biology elective. So I'm going to go into greater detail, a little more detail about this, and later, if required, I will post a video on genomic imprinting that I am recording for stem cell biology. So you can watch it there. So one chromosome from paternal, one from maternal. You can also have a situation like, I will call it as gene A, and this one is gene B. Gene A and gene B. So if there is a situation where A is expressed only from the paternal, and that means it's monoallelic, it should be repressed on the A allele of maternal A allele should be repressed. And it may also be like this, that paternal B is repressed and maternal B is expressed. Okay? So I hope you understood these three parts of the 3 parts to the definition of genomic imprinting. It is an epigenetic modification or way of controlling gene regulation. It is monoallelic expression. That means only one of it is expressed, and the expression is dependent on the parent of origin. Some genes are expressed only from the paternal allele, like in this case A. Some genes are expressed only from the maternal set, like in the case here it is B. Okay, just that much you remember. We'll get to it in a while. And I hope you all know Southern blotting. And we have, in this course itself, we have discussed a lot about restriction endonucleases, and that is why because we are going to use restriction endonucleases for this particular technique, I am taking this at this time, because we have just recently studied restriction endonucleases. And the important part of this lesson is your ability or reasoning. It's simple like Minesweeper, or your reasoning and your ability to deduce the answer, or your ability to predict an answer. Okay, you all are supposed to also take a paper and pen, note down some of the points, because in the last 3 slides you are going to solve problems, and then they are incrementally, they will increase, they will have increasing complexity, and I want you to follow them properly. Okay. Yes. So genomic imprinting can be made. It is an epigenetic modification. It could be of DNA methylation or covalent or non-covalent ways of DNA protein interaction or DNA RNA interactions. But the best understood ones are about DNA methylation, and that is what we are going to discuss today. I hope you know what are CpG islands. Right? I think, did we discuss this, or haven't we? Or maybe not. Just let's go ahead, have a small discussion on what are CpG islands. So in eukaryotic, or especially in mammalian ones, promoter regions, or not all promoter regions, some of the promoter regions are highly rich in this region. If I have to blow it up, they are rich in CG, like this. You have multiple copies or repeats, CG repeats. Okay, they can span in hundreds or so, sometimes even thousands. So those are called as CpG islands. C is for the cytosine, and P is the phosphate bond between the phosphate backbone between the C and G. You can just say CG, I think. So these CG islands are highly prone to methylation. There are a lot of enzymes, DNA methyltransferases, and so on. They can methylate the DNA on the cytosine. The 5th position of the cytosine is methylated. So we are going to read that particular part. If we want to study genomic imprinting, or even if it is epigenetic ways also, this becomes very handy. So I chose genomic imprinting so that I can introduce you to this complex phenomena, and also the problems. When you get to the problem, you will understand. You will have complex way of thinking, and that is what I would want to. I hope you understood what is genomic imprinting basics, and the CpG islands methylation would happen. So here we are going to see one example. Here is an enhancer. To the enhancer, activators are bound. Okay, and there is a gene called H19, and there is a sequence called as insulator to which a protein binds. For this specific protein here that is bound is called as CTCF, and there is another gene called as IGF2. Okay, there are two genes, two regulatory elements, enhancer and insulator. Enhancer binds to activator, and activators are important proteins that are responsible for activating transcription from certain promoters. So activators will allow the formation of mediator complex and other promoter transcriptional apparatus to allow transcription to happen. So usually this is the promoter region, and you may have the activators that are shown here will interact with this, and then the transcription of this is enhanced. Okay, so for in this case, as you're seeing here, this DNA should have looped. This is a loop that should have formed. Only then the activator can interact with the promoter elements. Okay. So if there is also another possibility is that whether this DNA can loop or not is dependent on if this location is bound by something or not. If CTCF, the insulator is here, okay, this is the insulator. If CTCF protein is bound here, then it will prevent the looping. If CTCF is not bound here, then looping is allowed. Okay, I will get back again. So the expression of IGF2 requires the activators. Okay, if the activators should interact with the IGF2, then the DNA should have looped something as shown here. Okay. Whether looping is allowed or not is dependent on the CTCF that is bound on the insulator or not. Okay, let's finish off what happens in each of the cases, and then probably we'll be able to understand what we mean. So here is a situation where there are two parental sets. One is a maternal chromosome. I hope you remember that we have seen something here, right? Maternal and paternal like this. The same kind of setup, only with some more details. So this is a maternal chromosome. The individual has inherited from the mother. And here is a paternal chromosome, inherited from the father. So what happens in the maternal chromosome is there is an enhancer to which activators are bound. The activator can either activate transcription from IGF2 or H19, not both at a time. But by default, these activators would interact with IGF2. But for that to happen, DNA looping should have happened, something like this. But there is one problem, because the insulator is a sequence to which a protein called CTCF is bound. If the CTCF is bound, then looping is disallowed. Okay, so the enhancer would rather interact with this promoter and activate transcription of H19. So you'll have transcription H19 is on, but IGF2, the activator is not able to reach IGF2. So it would be turned off. Okay. Now let's look at paternal chromosome. What happens in there? You have the difference between paternal chromosome and maternal chromosome is one thing in terms of DNA. This part of the DNA is methylated. All the red ones that are shown, that is methylated. CTCF, so the methylation also spans from insulator region till the mid gene of H19 or so, approximately. So now you have a situation. Insulator is methylated, but CTCF cannot bind to methylated insulator. It can only bind to insulator that is unmethylated. So now the CTCF cannot bind. Then what is allowed? If CTCF is not there, then looping is allowed, right? Because there is nothing physically obstructing the looping. You will have the interaction, because of which you will have the transcription of IGF2. Now, that is especially because the activators are able to interact with IGF2. So IGF2 is on, but H19 is off. Why is it off? There could be two reasons. One is it is methylated. This region is also methylated, which includes the promoter of H19. If it is methylated, the transcriptional machinery may not be able to interact with this region. That is one reason. The second could be that the enhancer is now busy kind of activating, because it has looped and is now interacting with the promoter complex of IGF2. It is not available for activating transcription from H19. I hope you understood something here. What are the three things that I said for genomic imprinting? First thing, it is a epigenetic mechanism, right? How is it? Why is it an epigenetic mechanism? Because it is involving methylation, right? It is DNA methylation is a type of epigenetic regulation. Second part is, I said it is about monoallelic expression. Okay, so if you notice that from the maternal chromosome, you have H19 produced, but IGF2 off. And if you see again in terms of, okay, I have to redo this, 1 second. Yeah, I wanted to describe just the monoallelic expression. There are two copies of H19. Here is one copy and here is another copy. Even then, there is only expression from one and the second one is off. That is monoallelic. Only one copy is expressed. The same can go for IGF2. IGF2, you have two copies, but only one of it is expressed. That is monoallelic expression. The third part is expression should be dependent on parent of origin. Okay, parent of origin in the sense here it is about whether which is expressed from paternal chromosome and which is expressed from maternal chromosome. Here from H19 is always expressed from maternal chromosome and IGF2 is always repressed on the maternal chromosome. IGF2 is always expressed from the paternal chromosome and H19 is switched off. So it is following the parent or the sex of the parent. I mean, the expression pattern is dependent on the parent of origin. So these three things are the prime parts of genomic imprinting. Okay, if you need, you can always watch more YouTube videos, or I'll try to post a video when I make one. But this is simple. We don't exactly understand why such a system has to evolve. But if you have studied uniparental stem cells and other things, it is thought that it will prevent the fusion of, say, I will just give a small introduction to that so that you will probably understand the significance of genomic imprinting. Say, we will take the same situation like here. If two sperms fuse, okay, this is having one sperm is come with 1N haploid set. Now you have a cell with 2N. Can it give rise to an organism or not? Usually they grow to early embryo stage and then die. You can think of why that situation is happening. Say, now look at these chromosomes. I said dependent on parent, right? How many copies will be expressed of IGF2? Both are coming from paternal. IGF2 is expressed, okay, and IGF2 is expressed. You have twice the IGF2 required. And what about H19? You have 0 H19. So these organisms die out early. You can also think of the same thing in terms of an egg. Egg is here and it is undergoing cell division. It only, for some reason, you have activated it into a zygotic state. So it is having 2N. And the 2N, now if you see the chromosome sets, you will have 0 IGF, because both are inherited from the maternal. Both are IGF2 will be 0, whereas H19 you'll have twice the copy, and that results in a kind of abnormality, and the zygotes will die. So one of the explanation is that genomic imprinting can or prevents this kind of parthenogenic type of growth or asexual reproduction. But evolutionarily, we have to still think upon it and read more to understand the relevance. Or probably it is still unsolved. Maybe when you try to find out, you can, when you work on it, maybe you will find out. Okay, I hope you understood a little bit about genomic imprinting. There are many other genes which I'm not going to discuss, okay? There are several hundreds or thousands of genes, and the list is increasing each day based on the organism that is studied. This is the next one that is important. I want you to note down the things that you are hearing here, okay? So here is an organism, and we are talking about imprinting. What happens during the life cycle? When is imprint made? This individual has two sets, right? One are somatoplasm, and it also has the, within the gonads, it will have the germplasm. So somatoplasm, all of it is, one is inherited, say, from the father, and the other one is inherited from the mother. Okay, one is a paternal chromosome or genome set, and another one is a maternal genome set. The black one indicates that it is methylated or not expressed. This is not expressed here. You have a monoallelic expression. You have two copies of these two. Here is one copy and here is another copy, but only one of it is inhibited and one is expressed. The same goes for these two genes. There are two copies of the same gene, but one is methylated and the other one is expressed. Okay, in germplasm, something happens. It's called imprint erasure. Erasure of imprint, whatever you want to say. So what I mean to say is, when the germplasm in the early embryonic stages, the germplasm is formed from primordial germ cells. So the primordial germ cells first form, which will eventually give rise to gametangial stem cells, and then they will give rise to gametes. Okay. So the primordial germ cells, when they are forming in the early stages, all the imprints are erased. Say, for example, here you have, here you had methylation. I'm trying to put it as the black one I should put. 1 second. So I'm trying to put what is shown here in this case, and then tell you what is going to happen. So this is the one gene that is methylated, so it is repressed, and the other gene is here, it is methylated. Okay. In primordial germ cells, that is, when I say this is imprinted, it means it is actually methylated. So it is repressed. Erasure means all of it will be demethylated. They will erase it. Now there is no methylated DNA as a part of imprints anymore. So that is called as imprint erasure. We will talk this individual as a male. Okay, this individual, the embryo is going to become a male in the future. So primordial germ cells you have erased, and we are not considering this part. This usually comes from a different organism, right? If it is a male, it will produce sperms like that. So imprint is erased, then it will form gamete. When it is going to form sperm, the imprint is established. So here is the imprint is established. That means what should it imprint? I will simply give it as a situation like this. Probably you can then try to answer, see what you can. So from one spermatogonia, you will eventually have four spermatids, okay, upon meiosis. Just for understanding, we will put it like this. And there is a, they are all N, that is haploid. So they will only have one set of the genome. Now, if you think of these genes, should there be any methylation or should there not be any methylation? This is assumed that the primordial germ cell, primordial germ cell is here, and that gave rise to spermatogonia, which will eventually give rise to sperms. So by that time, from formation of primordial germ cells, all of the imprints are erased, and then we have an establishment. If you can see in this case, I said this belongs to the paternal and this belongs to the maternal set. And I also said that this individual is a male. What would you expect then? The imprint that all the cells that are coming from this particular cell should be something like this. All of them should have only one type of imprint based on the sex. Okay, and eventually you will get eggs from a different organism, and then they will fuse to form a zygote. And then whatever has happened, it will be maintained in this embryo throughout the somatoplasm. What happens in the germplasm of the new organism, this cycle will keep on running. So what you mean to say is, generation after generation, what we will see is that imprints are erased and imprints are made. How you can think of is like think of yourselves as an individual. The imprint that our parents have made, our body is made up of imprints that we inherited from our father and mother. They have made the imprints in their gametes, and that's what is our somatoplasm. When the next generation is about to come, you would have in the gametes, you would have made an imprint, and the partner would have made another imprint, and the child will inherit those imprints. Okay, epigenetics and genomic imprinting is a fantastic area of research. I think I have shared you one of the Coursera course about epigenetic gene regulation. If you are interested, you should try to finish that course. It's a fantastic one. Okay, what I want you to remember for this, for the rDNA perspective, is what are imprints? When does erasure of imprint occur? That is during the formation of primordial germ cells. When is imprint established? During maturation of gametes. That is what you should remember. And when we try to solve the problems, you will get a better understanding. Hang on and try to follow what is there. So in epigenetics, in the next, we are going to check only this part, okay? I hope you all know about Southern blotting. So I'm going to quickly review it, what you would do. You have, what is the objective of Southern blotting? It is to find a gene of interest or a sequence of interest, whether it is present or not within a DNA sample. So you would have isolated a DNA like this, and then the objective is to find out if this gene A is present or not. Okay, so what would you do? You would have DNA sample, then you would have restriction digestion. You want to have it in manageable fragments, so you would have restricted digested it. Then we would have done electrophoresis. Once we have done electrophoresis, the DNA would move based on the size, smaller faster, and larger ones slower, and you would have size fractionation, something like this. And then what we do, I'm continuing here. Okay, so here is the electrophoresis gel. We can't know, even if if this assume this is where the gene A is present, there is no way of not identifying that is the gene A, because all of it is DNA. There is nothing to indicate that is the gene A. So we should have used some probe with a radio label or some other label to identify that is the gene A. So you have electrophoresis we have done. Now we will do blotting. We will put a nitrocellulose membrane to which the DNA will stick, because the buffer moves up, and the DNA will get stuck to the nitrocellulose filter. So we will peel off the nitrocellulose filter and put it in a tray containing the probe. I hope you remember in one of the lectures we designed a probe, and I also discussed about in vitro transcription and how to randomly label a probe. The same thing applies here, but we will discuss more at a later time. So assume a probe. What is a probe? First thing is, a probe is a nucleotide. Here in this case it is a nucleotide that is complementary to the sequence that we are looking for. So it should have exact match within, or majority of the things. There should be homology between this is the probe and the probe. The second property of the probe here is it should have a label. Preferably it is a radio label, but there are also others that are available. But that's what it is. So probe is a nucleotide which is complementary to the region of our interest. Here I said gene A is our interest. Then when we add the probe, it will stick to the bands whichever have the gene A sequence. And then once you develop the radiograph, you will find out the bands that are present. Whichever bands are there will form a dark band. So quickly, DNA isolation, restriction digestion, electrophoresis, then you have blotting, then probe hybridization, and then detection. Okay. And then I will quickly move through these. You also have learned about restriction endonucleases. Some of them are methylation sensitive, some of them are methylation insensitive, and some of them are methylation dependent. So the question here is, here are 3 examples, and why we are looking at these is important. We have chosen NOT1, HPA2, MSP1. Why is that? Because we are talking about DNA methylation. Epigenetics, in the epigenetics, we are trying to analyze the DNA methylation status. And DNA methylation usually occurs on CPGI lines. That means there are several CG repeats like this. So we have chosen restriction enzymes that have CG-rich target sites. NOT1, it is an it recognizes an octamer. Its sequence is GCG, GCC, GC. HPA2, it's a 4-base recognition sequence. It recognizes 4 base. CCGG. And MSP1 is also CCGG. But there is one difference. That is, if you look at these two are methylation sensitive. What does that mean? NOT1 can cleave this sequence only if it is unmethylated. If the target site is methylated, it cannot cleave it. The same is the case with HPA2. It cannot cleave methylated target sites. Its target site is CCGG. It cannot cleave if this sequence is methylated. MSP1 is special. It is methylation insensitive. It is the HPA, and I will just take this here. HPA, HPA2, and MSP1. If it is, I'll also maybe CCGG. And I'm putting this one as methylated. There are two recognition sites, CCGG and CCGG. MSP1 can cleave this one, but cannot cleave methylated strands. If we take MSP2, it can cleave unmethylated and it can cleave also methylated target sites. So there is only one difference between HPA2 and MSP1. HPA2 is methylation sensitive, whereas MSP1 is methylation insensitive. There are also other restriction enzymes such as DPN1, which will cleave only if the target site is methylated. We are not going to use this for this technique, but we're going to employ methylation sensitive and methylation insensitive enzymes. So from now on, please take a paper and try to answer as soon as possible. Here is a setup. I want you to answer. This is the DNA. Okay, DNA is assume this is the genomic DNA, and you have from here till here. This is an EcoR1 site, and here is an EcoR1 site. I think I will solve this one for you. And here is a NOT1 site, and here is HPA2 or MSP. They recognize the same sequence, right? CCGG. Only difference is about methylation or not. We are not talking about methylation and all this stuff now. So this is how it would look like. This is the distance between these two is 3 kb, and N and H, this NOT1 and HPA2 site is 1 kb, and EcoR1 and NOT1 is 2 kb. If I performed DNA isolation, restriction digestion just only with EcoR1, electrophoresis, blotting, probe, and then autoradiography, what am I likely to find? Where would I find a band? You can tell. Please answer 1 kb, 2 kb, 3 kb, 4 kb, 5 kb, 6 kb. The total size is from EcoR1 to EcoR1. What is the size? It is 6 kb. And here is the probe. Okay, I didn't mention about the probe, right? Probe is homologous to this particular region. Okay. Now I will try to solve this, and then you will probably understand what I meant. So if I digest just with EcoR1, EcoR1 would cut here and here, right? And then I have added the probe. Probe would bind to this fragment here, around this place. This total size is 6 kb. So what I would find is a 6 kb fragment like this. A band representing a 6 kb fragment. Now, if I treat it with EcoR1 plus NOT1, what would happen? EcoR1 would cleave in this location here, and it will also cleave here, and NOT1 will cleave here. What would I get then? This is 6 kb fragment. Okay, I will draw a line. That is a 6 kb fragment. Okay, but then that is with EcoR1 sites, but then NOT1 has cleaved it in between, something like this. And where is the probe going to bind? Probe is going to, you see here, NOT1 is here. Probe is going to interact with this one. What is the size of this fragment here? This is about 2 kb, and the rest of it is about 4 kb. which band will be visible on the gel? Will this one 2 kb fragment is visible on the X-ray film, or the 4 kb, and why? Yeah, you're supposed to answer.
Only the 2 kb fragment will be visible.
Yes, louder, please.
Only the 2 kb fragment will be visible.
Why is that?
Only because it's the one where the probe is attached to. So
That's right.
one will be.
Yes, probe is homologous to this fragment, as you're shown, as you're able to see it here. Okay, so it will only that band will be recognized. The 4 kb and rest of the any DNA will not be visible. What about E and H? That is EcoR1 and HPA2. EcoR1 will give the 6 kb fragment, something like the previous one we have done, 6 kb fragment. But then we are digesting with HPA. Yes, Shubhakirti?
3 fragments, sir.
3
kb.
Yes, that's right. You're right. So HPA2 is going to break it here, right? Okay. So HPA2 will break it here. What are the sizes now? You can see it. This is 3 kb because its cleavage is here. So from here to here, it is 3 kb, and the rest of it is 3 kb. But only the probe is placed in this location, so that fragment will be visible. What about EcoR1 and MSP1? Assume that this DNA is unmethylated. Okay. What would you find? Anybody? You would find the same, because MSP and HPA, they cleave at the same location, isn't it? They will form a 3 kb fragment and a 3 kb fragment like this. If anybody is losing out, don't worry about it. We will discuss these problems again, but it is a very good exercise if you can follow it now as well. Please solve this problem now. I have changed something. I added a complication to this. I changed this. The probe was previously present here, but now I have changed in a new experimental setup. I made a probe that is going to interact, that is homologous to these regions here like this. Okay, let's go on. With EcoR1 alone, what would I get? We will find a 6 kb fragment. EcoR1 plus NOT1. So NOT1 is going to cleave here, and so this 2 kb fragment will be cut off. The leftover is 4 kb, and the probe is binding to this fragment. So we will find a 4 kb fragment. What about EcoR1 plus HPA2? What would happen there? EcoR1, and so you have 3 fragments. This is 3 kb fragment, and this is another 3 kb fragment. So because these both are of same size, they will form only one band like this, 3 kb fragment. EcoR1 plus MSP1 also is the same situation. Okay, so that is how we need to keep thinking. So next one, I'm going to have an additional complexity. Here is, now, as we know, all the mammalians are eukaryotes, right? If you're trying to do epigenetics, then we have to see that we should notice that they are diploid, and they are biparental. So I have the same setup, except there is only one addition I have made, is that this region is methylated. Okay, everything else is same, except you have one paternal copy and a maternal copy, and in the maternal copy you have a methylation. So what would you find in that case? With echo R1, we'll have the same banding pattern that was just put to echo R1 plus not 1. What bands will form? And you should be able to tell me that now. Anybody? So in echo R1, both these will form a 6 kb fragment, right? And then what happens? We are using not 1. What does not 1 do? Not 1 is methylase sensitive. Okay, methylation sensitive means if it is methylated, it cannot cleave. If it is not methylated, it can cleave. So in the paternal allele, it is not methylated, so it will cleave. In the maternal allele, the region is methylated, because of which it cannot cleave. And the probe is going to bind to this region, right? This is 2 kb, and this one is 4 kb, and this is 6 kb. What would you find then? The banding pattern. You will have a 6 kb fragment, and you will have a 4 kb fragment like this. What about E and H? E and H, it's easier to draw this way, and that will help your understanding better. So E and H is going to cleave in the middle like this, but H, that is HPA2, is methylation sensitive, so it cannot cleave the other allele. So what we'll end up is with a 4 kb fragment. The probe is binding here and here. The other one is a 6 kb fragment. Okay. Are we doing it right? No. Sorry. This is a 3 kb fragment. Sorry. HPA2 cuts in the middle, right? So it is a 3 kb fragment and a 6 kb fragment. So you'll find one 6 and another one is 3 kb fragment. The last one is E plus M. I will just draw again. Here are two DNA molecules, and one is methylated, right? I'll put it as this is methylated, the other one is not. MSP1 does not care if it is methylated or not. It is methylation insensitive, so it will cleave both like this. And now you would only get a 3 kb fragment. You will not see the 6 kb fragment like you have seen here. Is that understood a little bit?
Excuse
Yes, please.
Sir, in the unmethylated one, will
Yes.
the probe will be able to attach with it if the strand is broken? Because first you are resisting it and then the probe is coming.
Say it again once.
So this probe
Mhm.
will be able to attach it to the strand since it will be broken with P1
Okay,
and
I
SP1.
understood your question. So you're meaning it is broken, so how can one probe bind to both? Is that right?
Yes, sir.
Something like that? Yeah?
Yes.
So if you have a DNA like this and it has been broken in pieces, this is the base, and the actual probe design is something like this, right? And when you do electrophoresis, these would have been in 2 different places like this. Say, okay, this is part A, or sorry, this is A and this is B. A is here and B is here. How would the probe bind? You have multiple probes, and as long as the probe can form base pairing like this, then one probe will bind to it like this, and it will form base pairing like this. In a different scenario, in case of B, another probe will bind like this. Okay. Did I answer your question?
Yes, sir. Thank you.
Is that what you asked, I think?
Yes, sir. Yeah.
Yes. Okay, so it's not a problem. As long as there are sufficient number of hydrogen bonds that a probe can form with the target sequence, the multiple probes can bind like this. That is how you will end up with 2 bands or 3 bands. Okay. I hope you understood this one, and I think we are running out of time. Please take a snapshot of this screenshot, and I'm going to explain the problem. You are supposed to answer it and upload it on our WhatsApp group. Okay? Here is everything else is same. I'm not changing anything at all. I'm only adding a complication now, is that one sample belongs to the somatic cells. Okay? This you have to analyze. You know the imprinting status, what happens. From somatic cell to primordial germ cell, erasure occurs. From primordial germs to formation of mature gametes, you will have establishment of imprints. Okay? And imprints can be two types, either maternal or paternal, like is shown already here. So based on that, you have somatic cells. We have isolated DNA from somatic cells and analyzed this way, right? We have isolated DNA from primordial germ cells and also analyzed the same way. Similarly, assume we have isolated sperm. Those are mature gametes. What we have analyzed with restriction digestion, southern blotting, everything similarly. And here is the egg. Same case. What would be the banding pattern in each of these cases? This problem might come up in the exam. That's why I'm spending time. It is a good problem because it will allow you to understand things in a complex way. When you read a research paper, for example, in this course, we are also going to read 5 or so research papers. If you want to really read a core biology research paper, one should be able to associate a lot of knowledge, factual knowledge, with factual and conceptual knowledge along with your interpretational skills. That means we are increasing the complexity. I hope you realize that here in the first one, we just did with one and a probe we have taken just here. And in the second case, we have taken a probe where 2 pieces could be identified by the same probe. And in the next one, I increased the complexity by adding 2 sets, that is diploidy. I included that, where methylation also comes. And then finally, we have the whole developmental stages, at least the germplasm developmental stages. In all these cases, we should be able to, the point of this whole exercise is your ability to think critically in complex situations. So I'll be waiting for your answers. Take a snapshot of this. You can digitally mark the bands on it, or even if you do it on paper, I would like that. And did you all take a print screen of it? And then save it for yourselves. Then you can upload the answer in the WhatsApp group is fine. Okay, I'm just trying to make it easier for everybody. So if you have any questions, you can also post so that next time when we meet, we can discuss upon that. And if there is any lacuna for anybody, if anybody wants to have a clearer understanding, I would be able to then customize it in that way. So please post your questions. You can post it on WhatsApp to me, and then I will try to customize the next talk based on that. Okay. Today has been a long day, so I think I will let you go now. Please try to solve it and post it. I'm waiting for your answers. Okay. Thank you very much. If you have any questions, you can ask.